Zoology, we're not doing lab today because we don't have time going over the tests and doing this here. I want to catch up on this stuff because I do want to have one more lecture exam 3. I want to have that before we go away for Thanksgiving break. So I want to do our lecture exam 3 on Thursday the 21st. Work for you? Yeah? Everything?
That's so soon.
November 21st. That's a little over 2 weeks away, right? So the reason why is when we get back from Thanksgiving break, we only have 3 weeks left in the semester. And we got to cram in a lecture and a lab exam. So those are going to be short.
Oh no.
So anyway,
maybe it'll be fun.
So
maybe there
the
won't
positive
be
of all
stuff to
this,
make it
the
bad.
plus is that
Because this one was kind of bad because it was a lot of stuff.
Like 2.5 weeks to go over the next content, it's not going to be as long of an exam, it's a shorter exam. So it's going to be a little bit lighter
points.
exam. And then the other positive is over Thanksgiving break, you don't have to worry about remembering anything from the lecture exam. Just go away in your heads. So our next lecture exam 3, is going to be on Thursday, November 21st. Right? So that means no lab that day, just lecture exam that day. Then, when we get back from Thanksgiving break, which is this whole week here, we have the whole week on, we only have 1, 2, 3 days. So what we'll do is we'll have a lecture exam on Tuesday, and this is going to be a lightweight lecture exam because we only have like 2 weeks to cover stuff, and then we'll have a lab exam on the next day. And the lab exam on this one, I think it's pretty straightforward because it's all big stuff. We're going to basically be doing mostly vertebrates because they're so big. I think people do the best on that one because they're kind of more familiar with what the frog has versus what crayfish has. There you go. Okay, so that's our plan. Unfortunately, Thanksgiving break this year was.
I was so grateful that all of our exams weren't doubled up, but those are.
Come back to lecture exam 4 and lab exam 3 last week. And I just piled up with all kinds of tests. That was for sure. Okay, so this is content that we're going to finish up with physiology before we jump into tissue physiology. So let's go ahead and cover this. Is arthropods and colonization plane. So there's some pretty complicated stuff I want to go over on this with regards respiration. And so let's take a look at kind of like the aquatic or marine versions of these arthropods, and then we'll look at the terrestrial versions and how they work. So for the chelicerates, the cheliceratas, so what we're going to do here is kind of like a compare and contrast. How do the marine versions do respiration versus how do the terrestrial versions do respiration? So for our chelicerata, the chelicerata, which includes the horseshoe crabs as our marine version, what we have going on here is the horseshoe crab, for example, has a body. We have our little appendages here, and like this, okay, 1, 2, 3, 4, 5, 6, and then they have a histosoma that has these plates on the histosoma. So we have these book gills. And they're called book gills because they kind of look like if you have a book and let the pages kind of flop like that, the book gills kind of resemble the pages of a book where that name comes from. So this is where we have respiration occurring, oxygen will be absorbed by diffusion, and CO2 is released by diffusion. And these organisms basically are in close contact with the water, so they're essentially doing respiration on their book gills. Got it? Pretty easy. When these chelicerates moved onto land, for example, with scorpions, let's go ahead and do draw the scorpion here. This. Scorpion legs. Okay, so here's our scorpion. This. Scorpion with a little stinger on them. Okay, and so with the scorpions, what's happened is the book gills have been pulled into a cavity. I'll just draw two, even though there's actually four in scorpions. They have 4 pairs of book lungs. Okay. And what they did basically is they have the same structure, these book gills, but they pulled the book gills into these pockets that we now call book lungs. And the benefit to that is that the respiratory plates are no longer openly exposed to the atmosphere, because the problem with living on land is desiccation. So because the air dries the organism out, especially respiratory surfaces, having your gills kind of hanging out and exposed is risky. And pulling them into a little cavity where you can open and close the little opening to the cavity, little slits, allows these organisms to prevent water loss and regulate their respiration. So they're able to do respiration, but they're using the same structure as the marine version. It's just they're pulled into a pocket that can help them retain water a little better. Got it? That's kind of the story there. So here we have horseshoe crab. Versus scorpion or spider. Okay, so that's pretty easy. All right, and then if we look at crustaceans, let's go ahead and now we're going to look at another group here, crustacea. So crustaceans traditionally have a gill off of their leg. So here we have, here's our crustacean here, like a crawdad or a lobster. Okay, so we have a bunch of appendages. And of course have the jellyhead, so you know it's a lobster. Gotta have 5 legs, 5 pairs of legs. And then you gotta have swimmerets. Okay, so here is our crayfish or lobster, and the base of the gills had these base of the legs. I'm sorry, had these gills that were suspended off of them. So here we have. These are just called plain old gills. I'm not sure why the gills on the horseshoe crab are called book gills and the ones on the crustaceans are just called gills. They don't have a special name. That's probably the history of the whole thing. But we just call these gills. They're essentially branches off of the walking legs that are used for respiration. That's their thing. So crustaceans have colonized land, and there's a few examples. There's not really a huge diversity of them, but we have these things that are called sow bugs or roly-poly bugs or wood lice, a whole bunch of different names depending on what country you're from. But these are little armored crustaceans that live on land. And they have, go ahead and give them some appendages. They're kind of all walking legs here. Okay, so the fun part though is the very, very hind plate here has gill plates. It has actually gills that are at the base of what would be the abdominal legs. So the gills are basically here at the very, very end of the abdomen. But of course, I mentioned that there is a big risk of drying out for these organisms. And so what they do is these wood lice or roly-poly bugs or sow bugs, sometimes they're called, they live kind of in damp environments. Usually in the morning they have to go out and find a drop of water somewhere, like a dewdrop of water or a raindrop of water. They're kind of in moist environments. And what they do is they collect a drop of water. They put a water drop over their gills. So in a way, they are bringing the water onto land with them in order to do respiration. They're keeping their gills wet with a drop of water, covering their gills, and that's where their respiratory surface is. So they do their respiration basically through their through this drop of water. So this is why these sow bugs, or roly-poly bugs, or whatever you call them, we'll go ahead and call them roly-poly. Hold on a second. Sow bugs. In England, they call them wood lice. But anyway, these are terrestrial crustaceans, and they're doing respiration by bringing water up onto land with them and covering up their gills. Okay. So it's kind of a different strategy. So it's likely that the chelicerates were the first to colonize land using that method. Crustaceans probably were the next group to colonize land using kind of like a modified gill in this way. Got it? Okay, and then that kind of leads us to our hexapods, or insects. So if you go with the idea that hexapods are actually kind of a modified, what do you call it? It's a crustacean ancestor. Crustaceans and hexapods probably had a common ancestor. And I think I showed you this, what do you call it, jumping bristletail or silverfish is an example of a really primitive hexapod that has a lot of crustacean features. But an alternative strategy for doing respiration. So we have kind of the crayfish moving on the land as a roly-poly bug. But the alternative idea here is the crayfish, or the crustacean ancestor, also lost its gills and developed these tracheal systems. So if you look at, say, for example, like a cockroach or an ant here, that some legs on them, hexapoda. Okay, so this is our hexapoda, or insecta. Same kind of thing. And what they've done is, rather than have a gill, they have these spiracles that are lining the abdomen and the last two segments of the thorax. And the spiracles radiate into tubes that go throughout the inside of the insect. And this is what's called the tracheal system. The cool thing about the trachea is it's incredibly efficient. The reason why it's efficient is the oxygen never has to dissolve in blood. So in these organisms, they have hemolymph that the oxygen dissolves through the gill membrane. It's transferred to the hemolymph, which has hemocyanin, a blood pigment, and the hemocyanin carries the oxygen to the tissues, and that's great and everything, but we have basically the blood is kind of like the slowest point of respiration. Whereas in the hexapods or insects, the oxygen goes from the atmosphere all the way down to the individual tissues as a gas. It never dissolves into the blood. So hexapods, insects for example, don't even have hemocyanin. Their hemolymph is purely for nutrient and waste circulation. It's not for respiration at all. They take the atmospheric oxygen all the way down to the individual tissues. Okay, so on the test, there'll probably be a question about, describe 3 ways arthropods have colonized land. And they probably colonized in 3 different ways. It was probably the chelicerates first, probably the crustaceans second, and then probably an additional colonization of crustaceans as hexapods third. And so there's essentially 3 different ways that arthropods breathe on land, right? Each way basically prevents water loss and maximizes respiration. But let me show you why the tracheal system is probably the most efficient, and that has to do with how much oxygen is available in the atmosphere versus what's available in the water. So I know this is a really complicated table, but what I'll try to do is share with you exactly what I want you to say on the test, right? So let's check this out. When we look at the atmosphere, if we look at atmospheric gas, it's actually there's a mistake here. This should be milligrams. Let me fix this real quick. Fix this real quick here. This is not grams, it's milligrams. So okay. All right, there we go. Okay, so the atmosphere basically weighs about 1,000 milligrams per liter. So basically, that would be like, if we said it was grams, it would be 1, 2, 3, 1.2 grams per liter is what the atmosphere weighs. Versus water has a much higher density at about 1,000 grams per liter. We're looking at fresh water, for example. So we're looking at a big difference, almost like 1,000-fold difference in the density of the material. Air obviously is less dense than water, it's pretty obvious. So if we look at atmospheric gases, 78% of our air is nitrogen. Almost 21% of our air is oxygen, about 20.4% is oxygen. About 1% of our air is argon. We talked about that with potassium-argon mating system. Argon is relatively inert, so doesn't really participate in this process. And a very, very small concentration of our atmosphere is carbon dioxide. 0.04% is carbon dioxide. If we were to translate that into actual mass here, that means that 995 milligrams per liter is nitrogen. 256 milligrams per liter is oxygen. We're going to ignore argon because it doesn't participate here. But a 0.5 milligram per liter is CO2. So the key feature here we're going to look at is, because nitrogen is relatively inert for a terrestrial organism, you breathe nitrogen in and it circulates in your system,
but it doesn't actually do any chemical reactions in your body. It just kind of comes in and goes out as it is. So we're looking really at the oxygen to CO2 ratio. So if we look at this ratio, 256 divided by 0.5 is about 500, right? A little bit more than 500. Does that work like that? If you divide by 0.5, it's like multiplying that by 2, right? That's when I check my math. So anyway, we could say that there is over a 500 to 1 ratio of oxygen. It's actually 512 to 1, but it's over a 500 to 1 ratio of, there's over 500 molecules of oxygen, or milligrams of oxygen, for every 1 milligram of carbon dioxide. And that's the number one thing about it. 500 to 1 ratio, 512 to 1 ratio, if you want to be exact, of oxygen to CO2 milligrams. So let's go over to water and see what happens in water. Most of water is actually water. Most of that mass here is the water H2O, which doesn't participate in respiration. Nitrogen has a very, very low solubility in water. Nitrogen does not dissolve very well in water at all. So if you look at how much nitrogen is dissolved in a liter of water, it's only about 20 milligrams of nitrogen dissolved in a liter of water. If you look at how much oxygen is dissolved in water, oxygen has medium solubility, meaning oxygen dissolves in water pretty good, but not as well as we think it does. There's only about 8 milligrams of oxygen per liter of water on average. And I'll come back to that in a second because it varies based on temperature. CO2 has a very high solubility in water. And CO2 can dissolve as basically carbonic acid and as bicarbonate. So carbon dioxide has a very, very high solubility. It can be essentially as soluble CO2 gas, carbonic acid, or it can be as bicarbonate, H2CO3. And so you can see that there's actually quite a bit of carbon dioxide dissolved in water naturally at 4 milligrams per liter. So look at the ratio here of how much oxygen to CO2. That's like a, what is that, like a 2 to 1 ratio, right? So if you're an organism that has to exchange oxygen versus CO2, it's based on diffusion potential. The idea is the greater the difference of those two gases, the more likely you're going to get oxygen transfer and CO2 released, right? So if the ratios are near equilibrium, it's pretty slow. But if the ratios are very, very different, it's pretty rapid. So a 2 to 1 ratio of oxygen to CO2 in water versus a 500 to 1 ratio of oxygen to CO2 on land, this favors exchange of oxygen versus CO2 on land, like a great magnitude versus in water. So one answer here is on land, it's far easier to exchange oxygen and CO2 because the concentration of oxygen is much greater than the concentration of CO2 on land, right? 500 to 1 on land, terrestrial, 2 to 1 water. The other thing to think about is just the overall abundance of oxygen. Look at this. There is the equivalent of 8 milligrams per liter of oxygen in water versus 256 milligrams per liter of oxygen in the atmosphere. If you divide those, 256 divided by 8, it's about 34 times. So what you can say is there is roughly 34 times more oxygen available in the atmosphere versus oxygen available in the same volume of water. So with 30, I think basically it's 32, I think. We'll just want to do the math. It's like over 30 times more oxygen available in the atmosphere versus in the water. So there's just more oxygen overall in the atmosphere. And there is way less CO2 relative to the amount of oxygen. So exchanging oxygen for CO2 is much more efficient. So this is probably one of the main reasons why animals evolved onto land is because of respiratory efficiency. It's easier to get oxygen in the terrestrial environment than it is to get it in the water environment. That, and there was probably a salad bar no one was eating. Plants colonized land, or plant ancestors colonized land first, and then animals quickly followed to take advantage of the food resource no one was taking advantage of. So, in addition to having a food resource in the beginning, there was a respiratory advantage of breathing atmospheric oxygen versus water respiration. And you can see this. Sometimes you can see, for example, in a warm pond, you can see certain kinds of fishes coming to the surface, grabbing a gulp of air and pulling back down. So it's really common if you have an aquarium, for example, that gets warm in your house. You'll see your little goldfish go to the surface and gulp air. Fishes can actually go to the surface and do bubble breathing, where they get atmospheric oxygen because the water's too warm. Let me add on to that in a water environment, the oxygen concentration is temperature dependent. So if you look at a curve of how much oxygen can dissolve in water depending on the temperature, cold water can be saturated with much more oxygen, almost twice as much oxygen than room temperature water can. So the 8 milligrams per liter that we mentioned, the 8 milligrams per liter that we mentioned, has to do mostly with room temperature water at 20 degrees, 25 degrees Celsius. As water gets warmer, it holds less oxygen. And this is one reason why we have, for example, certain fishes or certain aquatic organisms can only live in cold water. Trout, for example, trout or salmon need cold water because they need that cold water for oxygen transfer and respiration. If the water gets too warm, like if the water gets above 15 degrees Celsius in a stream, trout and salmon can't live there. But you have things like bluegill or bass that can live there because they can tolerate higher temperatures. So this chart here basically is talking about what types of organisms can live in saturated versus anoxic type water conditions. So the problem with living in water is, one, oxygen concentration is temperature dependent. Warm waters have less oxygen. That's one theory of what caused the Permian extinction at the end of the Paleozoic, is that the sea temperature increased because of climate change, and oxygen concentrations dropped significantly, causing a mass marine extinction. It also is one theory as to where lungs evolved from, is because when the temperatures decreased in the oceans, fishes potentially had to go to the surface in order to grab gulp air. And that's possibly where lungs originated. But we'll talk about that with rivers later on. The other benefit to living on land is there's just overall more oxygen available, 32 times more oxygen. And the ratio of oxygen to CO2 is much greater, 500 to 1 versus a 2 to 1 ratio. So the basic answer there is there's several reasons why breathing atmospheric oxygen, doing respiration on land is beneficial. There's 32 times more oxygen, and the ratio of oxygen to CO2 is 500 to 1. Versus in water, far less oxygen, and the ratio of oxygen to CO2 is pretty close, about 2 to 1. And so, but of course, the disadvantage of living on land is desiccation, right? Drying out, losing water to the surrounding atmosphere. But that's why terrestrial organisms tend to have a much smaller respiratory surface area compared to aquatic organisms. So the terrestrial respiratory surface area is much smaller compared to the aquatic respiratory surface area. All right, here's kind of a fun thing. So most hexapods or insects are terrestrial. And it seems that this tracheal system is an adaptation for terrestrial life. There are, though, a significant number of aquatic insects. Some insects have migrated their way back into the water. Less than 10% of all insect species are aquatic. Dragonflies are aquatic. Mosquitoes are aquatic as larvae. There's some aquatic diving beetles we'll talk about here. So what's interesting is we're going to look basically at how have the hexapods kind of evolved back into aquatic environments. So what I'm doing here is saying that hexapods or insects evolved from probably a gilled ancestor. They lost the gills and developed trachea. But then how do they move back into an aquatic environment? They've modified or an exaptation of their terrestrial systems. So if we look at living in the water, mosquito larvae, which I used on your exam here, mosquito larvae are an interesting example. They have a snorkel type attachment. They will basically live at the surface. Mosquitoes are typically filter feeders. And they live at the surface of the water. Here's a mosquito, for example. There's a number of other types of aquatic insects that do this. But since I put the mosquito larvae on your test, I'll show them. At the very, very tip of their abdomen, they have like a snorkel-like system that it's like a snorkel that oxygen and CO2 go in a breathing tube. And then when they have to dive underwater because a predator is coming or to get away from danger, they essentially just hold their breath. So they're going to the surface every couple minutes to get a breath of air and then going back down underwater. But essentially they're dependent on atmospheric oxygen using this snorkel breathing technique. So mosquito larvae, a number of hemipterans do this, water bugs, etc. They do this snorkel breathing. And that's one interesting way. But probably the most interesting, in my opinion, is the aquatic diving beetle and how it does its respiration. The diving beetle, it's a coleoptera, goes to the surface to collect a bubble of water. Oh, sorry, a bubble of air. They go to the surface of the water to collect a bubble of air. So I'll go ahead and draw a little beetle here. Okay, so the beetle goes to the surface, grabs a bubble of air, and then it will dive down and take that air bubble with it. So underwater, this same beetle here. Okay, so that underwater, this beetle has a air bubble it takes down with it. And of course, there is a tracheal system, like in the mosquito larvae, that transfers that oxygen to the tissue to get rid of CO2. So what I want to do is talk about the partial pressures and how this thing works. Because the general idea is, the general assumption would be that we have oxygen in here. We have mostly nitrogen in here and some CO2. So if you look at what the concentrations of these are in the atmosphere, N2, like I said, was about 995 milligrams per liter, according to our previous page. Oxygen is roughly 250 milligrams per liter, and CO2 is roughly 0.5 milligrams per liter. So in the atmosphere, basically we have these partial pressures are what's in the atmosphere. In the water environment, it's quite a bit different. You can see here that there's much less nitrogen in the water because nitrogen is not very soluble. There is medium solubility of our oxygen available at 40 milligrams per liter, and then there's a high solubility of CO2 in the water compared to the air. So what I've done here is I've shown kind of where these gases go. Nitrogen, because it's relatively inert, is not being used by the insect. But what's happening here is oxygen is being consumed by the insect. The oxygen goes into the insect and is being used for metabolism, and it's being exchanged for carbon dioxide. So the general notion here is it's kind of like if you were to go down underwater with a scuba tank, you're breathing the oxygen and just kind of exhaling the CO2, you're using up the oxygen in the scuba tank. But it's actually more complicated than that. What's happening here is the oxygen is being taken in by the insect and used for metabolism. The CO2 is being produced, but because the CO2 has a higher solubility in the outside of water as HCO3, the CO2 actually leaves the bubble as soon as it's made. So as the insect exhales CO2, the CO2 dissolves out into the water because it has such a high solubility in the surrounding water. Oxygen doesn't really have a very good solubility out here, so the oxygen basically goes into the insect. And what's interesting is because most of this bubble is made of nitrogen, the oxygen from the surrounding water actually comes in to replace the oxygen that the insect has consumed. So because oxygen that does not have a very good solubility in water, oxygen actually transfers into the bubble, even though it has a higher concentration in the bubble. It's relative partial pressure gases, basically because oxygen diffuses better in a gas than it does in water, oxygen diffuses into the bubble. So what's happening here is the oxygen's going into the beetle, CO2 is coming out, CO2 is released, but oxygen comes in to replace that partial pressure gas. Because most of the bubble, the vast majority of it here is made up, 78% of it is made up of nitrogen. The bubble basically remains or maintains its shape. What does happen is because the nitrogen slowly leaks out over time, it's a very, very slow diffusion of nitrogen out. What happens is the bubble slowly gets smaller and smaller and smaller, not because oxygen and CO2 are being exchanged, but because nitrogen slowly leaks out. So the only reason the beetle has to go back to the surface is not to get more oxygen, but it's to get more nitrogen. It's because the bubble size, the bigger the bubble, the more respiration transfer it's going to get. The smaller the bubble, the less oxygen transfer it gets. But if they were able to maintain the bubble's shape without losing nitrogen, the beetle would never have to go back to the surface. So this structure is kind of what's called a physical gill. This is called, this is a great test question. This physical gill is basically the bubble acts as a respiratory surface area, like a gill underwater, but it's not an anatomical part. It's actually just a bubble of air that operates like a respiratory surface. What I think is pretty far out and cool is this is like the opposite of what the roly-poly bug does, right? The roly-poly bug, in order to live on land, brings water with it. The beetle, in order to live in water, brings a bubble of air with it. But they're both doing the same thing. They're using the surface of that water, air transition in order to do respiration. That's pretty cool. There are some beetles that actually are able to hold that bubble of air under their hair on their abdomen, and they never have to go back to the surface. All they have to do basically is just hold that bubble of air to keep nitrogen from leaving. So the main punchline of the story here is that when insects or hexapods evolved back into water environments, they basically had to modify their terrestrial respiratory structures in order to live in the water, even though the original ancestor of insects were a marine or aquatic-based organism. They're not using gills in this case. So it's kind of a really cool example of like exaptation after exaptation, kind of like going full circle with your adaptation for respiration. There's a cool link if you want to check out that PowerPoint of how beetles breathe underwater, kind of summarizing what I'm talking about here. But on the exam, we'll have a test question. One of them will ask, describe 3 ways that arthropods, 3 different groups of arthropods have evolved onto land. And that would be scorpions and spiders, terrestrial crustaceans, and then terrestrial insects and hexapods. That'd be one question, probably for 6 points. And then a secondary question for probably 4 points would be describe how the diving beetle uses its environment to do respiration underwater. And the basic idea there is because of the exchange of gases between the atmosphere and water, CO2, because it readily dissolves in water, is replaced with oxygen in the bubble as the insect breathes in oxygen and releases CO2. The nitrogen basically stays pretty inert, but because it slowly leaks out, the beetle has to go back and get another bubble. But basically the bubble operates like a physical gill. So that'd be an important thing to say in your answer is the bubble is actually operating like a gill, but it's actually not part of the insect. That's why it's called a physical gill. Okay, any questions on that? So that's a good one. I gave you a good description there. You can pretty much what's on the PowerPoint is more accurate detail, but this is kind of the minimum story here. You notice that CO2 dissolves readily in water and oxygen transfers readily back into the insect. Okay? Okay, I think that'll be it for today. What we're going to do on Thursday is I want to finish up arthropod physiology by talking about how arthropods and nitrogenous waste. We'll talk about how malpighian tubules and green glands work. That's the rest of this PowerPoint. And then we're basically done with arthropod and invertebrate physiology. For this next exam, we're probably going to include in how muscles and neurons work. We'll look at neuron and muscle physiology. And so that'll probably get us all the way to that lecture exam on Thursday, the 21st. Okay? That'll be it for today. We'll let you go. And I'll enter those lab exam grades in right away. I updated the 2 people I corrected your lecture exam already changed your grade. So if you have any questions, let me know. But there you go.